Hello everyone and welcome to today's webinar, Unleashing the Potential of Multiparameter Selection Using the CGX10 System to Drive Next-Generation Cell Therapies. I am Cassie Saltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sony Biotechnology. To learn more, visit www.sonybiotechnology.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. Before we begin our presentation, we do have a few poll questions that we would appreciate your response on. You should now see your first poll question in the presentation component. This poll asks, how many immunological markers do you leverage to isolate cells of interest in your current cell and gene therapy manufacturing workflow? So you can select 1 or 2, 3 or 4, 5 to 8, or more than 8, and then click Submit to show us your response. We'll give you just a few moments to take that and respond to that first poll question. We'll give you just a few more seconds on this one before we move forward to our next poll. Okay, thank you for your participation in that first poll. Our next poll question here you should see reflected says, what stage in cell and gene therapy development process would best describe your current work? You can select either R&D stage, early process development or preclinical stage, clinical grade cell and gene therapy manufacturing stage, or all of the above. And we'll give you a little bit of time to fill that out as well. All right, thank you so much for your participation in that poll question. We'll move on to our final poll question before we begin. This last poll asks, what technology are you currently using for cell isolation in your current cell and gene therapy manufacturing workflow? You can select magnetic-based cell isolation, fluorescence-based cell isolation, combination of magnetic-based and fluorescence-based cell isolation, or other technology, acoustic, centrifugation-based, etc. We'll give you just a little bit of time to answer that as well. Okay, wonderful. I'd like to now welcome our speaker for today's webinar, Brian Gelfand, Field Application Scientist at Sony Biotechnology. Brian is a graduate of the Waksman Institute of Microbiology at Rutgers University, where he studied non-coding RNA function and transcriptomics. Post-grad, he worked at the Waksman Genomics Lab, contributing to genomic research in several species, including human, Drosophila, bovine, and duckweed. He then gained experience in flow cytometry and cell sorting combined with genomic applications at startup companies. Since 2020, Brian has worked as a field application scientist at Sony Biotechnology, specializing for the CGX10 Cell Isolation System. Brian, you may now begin your presentation.
Welcome, everyone. Today's talk, Unleashing the Potential of Multiparametric Cell Selection to Drive Next-Generation Cell Therapies, a deep dive into the Sony CGX10 Cell Isolation System's capabilities. I'm Brian Gelfand, Field Application Scientist and Product Lead with Sony Biotechnology, and I'm joined by Aditi Singh, Global Senior Product Manager at Sony Biotechnology. An overview of today's talk. First, we'll talk a little bit about Sony Biotechnology and our cell sorter and analyzer portfolio, including the CGX10 Cell Isolation System. We'll go into a bit further depth on this system, exploring the key benefits, features, and value. For the second half of the talk, we'll be looking at utilization of the CGX10 Cell Isolation System for three different cell manufacturing workflows. First, for Treg therapy, sorting of ultra-rare naive Treg cells directly from a leukopak, using the system's combination of enrichment and purity modes. In the second experiment, we look at the CGX10 and its use for CAR T therapy, sorting CD4 and CD8 positive T cells directly after RBC lysis in a mixed population. And last, looking at the use of the CGX10 for iPSC-derived cell therapy to enrich and purify differentiated cells prior to a formulation and removing the cytotoxic base cells. And last, we'll watch a short video on the system and talk about some other applications that we have on our website. Sony has been working in the life science industry since 2011. We produce a variety of cytometry instrumentation for research and development, for discovery, and now for working in the GMP space. This includes our line of spectral cell analyzers, including the SA3800 and ID7000. For research and development and isolation of specific cells, we make several different cell sorters. Besides the CGX10, we make the SH800, MA900, and FP7000, as well as the necessary consumables and reagents for cytometric experiments. This is backed by Sony's full commitment to produce and support high-quality, innovative solutions in this broad portfolio of different instrumentation for different applications. Today, we'll focus on cell sorters and how Sony has been able to expand the field of cell therapies development. The first entirely Sony-developed system was the SH800, released in 2012. This is a two-way, six-color droplet sorter with an automated startup and chip used as a nozzle. This line was expanded with the MA900 in 2017, a four-way 12-color sorter, and the FX500, a clinical and cell line-oriented sorter with removable and disposable fluidics. This robust portfolio has been used for a wide variety of applications in discovery, research, and development. The CGX10 expands our portfolio from just research applications into production of cell therapies in the GMP suite, building off of the lessons learned with all of these other products. The CGX10 is a completely closed system with a compact design, appropriate for use in process development and in GMP production of cell therapies of a variety of different types. The CGX10 Cell Isolation System benefits and value. It's a novel system which enables GMP-ready cell sorting for cell and gene therapy manufacturing. It has a unique proprietary microfluidic chip. So unlike droplet systems, there is no droplet stream, there is no electrical charging. All of the sorting action is constrained within the microfluidic chip. The system is completely closed, preventing contamination and allowing bag-to-bag sorting. It is what we describe as GMP ready, meaning that the instrument, the single-use kits, and the software are manufactured in the appropriate environments. They meet 21 CFR Part 11 compliance requirements. The system has a compact design, allowing it to fit easily into the GMP suite, and an easy-to-use workflow, facilitating scaling up for production. This simple operation also includes flexible user modes for process development, for manufacture and administration of data. While the system is running, it has a number of fail-safes, including automatic clog detection and recovery. It's an easy-to-use workflow, and it has a touchscreen-based interface, minimizing the amount of extra computers that need to be in the GMP suite. It achieves high cell viability by running at a lower pressure than other conventional sorters. There's no electrical charging of droplets, and it has active temperature control for both the unsorted and the sorted cells, which can be controlled independently to maximize the efficiency of any protocol. It generates reliable data using multiparametric selection, using optics similar to our existing sorters. It has 2 scatter channels and 8 fluorescence channels, allowing a variety of different antibody combinations to be utilized. It runs at a high speed, depending on whether one is running purity or enrichment. And in purity mode, it achieves very high cell purity. And just as an example, some data comparing the CGX10 to a conventional sorter. It has higher viabilities between 7 and 5 to 7%. And we will see this in further detail in some of the experimental protocols. When operating, the CGX10 offers 4 different sort modes for different types of applications, and we'll be looking at this in further depth and how this works in the second half of the presentation. Purity mode prioritizes purity of sample, runs typically at between 5 to 15,000 events per second, and guarantees a purity of 97% or higher. In custom purity, we use a balance between purity and efficiency for different applications. So we can go closer to this 97% purity, or if a lower purity number is required, such as 80%, we can run the system at a higher speed. Yield and enrichment modes prioritize yield and efficiency of capture. They permit some off-target cells to come along. These are useful for enrichment of rare populations from larger volumes and run at higher rates, 75 to 100,000 events per second. Looking at the different user modes the system allows, there's operator, which is intended for working in the GMP suite, process developer, when the system needs to be run more like a conventional cell sorter to build and develop protocols, and last, administrator mode for management of data and operators. In operator, the user is able to run sort operation with a pre-built SOP. These are 21 CFR Part 11 compliant runs that generate sort reports at the end for record keeping. The operator is also able to do the required monthly quality control on the system and manage the sort reports. Process developers can do all of these, but in addition, they can create compensation matrices for building protocols. They can develop an SOP running the system in an open mode, and then converting this to an SOP with the required instructions for the GMP operator. And they are also able to manage the data on the system. Last is administrator. They engage in both report and data management. They are also able to create the password or barcode protected accounts used by the other operators of the system. So this is an example of the graphical user interface with the multiple modes. After logging in with a password or a barcoded badge, which is included on the system, they are able to select the appropriate role for what's being carried out at a particular time. And each of these permissions is granted on a user-by-user basis. Utilizing the CGX10 Cell Isolation System for different cell therapy manufacturing workflows. So in this part of the talk, we're going to look at a couple of different experiments, some of which were run in collaboration with the Center for Commercialization of Regenerative Medicine, CCRM, in Canada. We're going to look at utilization of the CGX10 Cell Isolation System, first for Treg therapy and sorting of ultra-rare, naive regulatory T cells directly from a leukopak using a tandem enrichment and purity mode sort. Second, we'll look at the use of the CGX10 in CAR T therapy workflows, sorting CD4 and CD8 positive T cells directly after RBC lysis at predefined proportions of 50% each. This was using the system's custom purity mode. And last, we'll look at iPSC-derived cell therapy, enrichment and purification of differentiated cell types prior to formulation. So first, for Treg therapy. In this sort, we started with a whole leukopak and then wash and remove platelets on day 0. This utilizes the CGX10 and sterile tubing kit if a compensation matrix is being built from the pack. And at the end of day 0, what one has is purified leukocytes. At the beginning of day 1, the leukocytes are stained, a OP mode manifold is installed on the system, and then an enrichment sort is performed to get enriched naive Treg cells. These are then concentrated and reprocessed through the same manifold on the CGX10, since it's the same batch of cells, running a quicker purity sort to obtain highly purified Tregs. And then after resuspension in media, the cells are expanded for 2 weeks. At the end of that time, cell count, purity, and viability were checked, and the cells were also stained for FOXP3 and then analyzed on the SA3800 spectral analyzer. The sort results of this experiment are shown on the bottom of this slide. First, an enrichment sort was performed, processing 1 billion cells at 90,000 per second for a total sort time of just under 4 hours. 91% of the naive Treg cells were recovered, expanding the purity from 0.3% before sort and 22% post-sort. These cells were then concentrated and reloaded again into the same manifold, and a purity sort was performed, processing about 6,000 cells per second for a total time of just under 1 hour for the purity sort, and recovering 60% of the highly purified Tregs. These had a purity of 97% at the end of cell sorting, so the total sorting time to process the 1 billion cells is just under 5 hours. The unsorted Enriched and purity sorted cells were expanded for 2 weeks, and we see the results of that in the upper left portion of this slide. The unsorted and enriched cells only expand 2- and 3-fold, whereas the purity sorted cells expanded 14-fold. So we see the importance of having highly purified naive Tregs for cell expansion. We can also see in the graph in the middle that purity of the cells was maintained after the 14 days in culture, looking at the 2 rightmost bars on the graph. So we were able in this experiment to process 1 billion cells and obtain, after enrichment, a 99% pure sample. In addition to looking at purity and expansion, we looked at FOXP3 expression and 14-day viability of the naive Treg cells. In histogram on the left, we see that over 90% of the sorted, highly purified Tregs were highly expressive of FOXP3. When we look at viability numbers in the bar graph, prior to sorting, after the enrichment mode sort, after the purity sort, and after 14 days in culture, a 99% viability was maintained, indicating that the sorting process on the CGX10 Cell Isolation System is very gentle and not damaging to the cells. So in conclusion, an enrichment mode sort followed by a purity mode sort enabled isolation of highly pure, highly viable, expandable, and FOXP3 positive naive Tregs. The total time to enrich and purify for 1 billion starting leukocytes took less than 5 hours. And we find that enrichment mode is highly suitable for pre-purifying rare and ultra-rare target cell populations. Next, let's take a look at a CAR-T therapy workflow. Here, CD4 and CD8 positive T cells are sorted directly after RBC lysis at a predefined proportion of 50% each, using the CGX10's custom purity mode. So looking at this experiment, on day -1, a fresh apheresis was washed and RBC lysis performed on a CPAX. On day 0, the cells are stained for CD4, CD8, CD45RA, and CD62L. And then the system is run using the custom purity mode and running a protocol to produce a 1-to-1 mixture of CD4 and CD8 T cells. After the sorting, the cells are seeded, activated, and expanded for 7 days. And this is data that was generated in collaboration with the Center for Commercialization of Regenerative Medicine, CCRM, in Canada. Looking at the results of this experiment, at the bottom, we have the sort result. These cells were sorted in just under 5 hours again, starting with 6.6 E8 cells. The target percentages of CD8 and CD4 were 4.9 and 17.9% respectively, and we recovered a total of 4.7 E7 cells. So this is sorted at 40,000 events per second. We get a total purity for the sample about 90%, and we have a near 1-to-1 mixture of CD4 and CD8 positive cells. Prior to sorting, you can see it's about 3-to-1 with 50% impurities. This was able to be done on the CGX10 with no other additional instrumentation or reagents, resulting in a process simplification to obtain a 1-to-1 mixture of cells as required. In a different permutation of this experiment, we ran a multi-population sort from 2 different donors, looking at groups 1 and 3 on the bar graph. We see the pre-sort population is different between the 2 donors. We're trying to obtain an even 1:1:1:1 mixture of CD8 naive, CD8 central memory, CD4 naive, and CD4 central memory. What we see in the post-sort is most of them have about a 25% abundance with a 2% error bar, even though they started with different ratios of cells. So what this shows is that the purity mode with mixed ratio sorting is able to take variable starting populations, process them, and get a product which is even regardless of the start. In conclusion, the CGX10 has options for custom purity sorting, allowing target cell sorting at the required purity and scale for a variety of different applications. It can be also configured for sorting multiple target subpopulations at different fixed ratios for different types of combinatorial therapies. Last, we're going to look at iPSC-derived cell therapy, enrichment and purification of differentiated iPSCs prior to formulation. In this process, on day 0, cardiac differentiation is initiated, and that the cells will be dissociated and processed over a period of 10 days, switching from medium to medium, as indicated in the upper right. This process is highly variable. Typically, the cardiac cell marker expression will peak between day 8 and day 10 of differentiation. There is high run-to-run variability. Another caveat to this protocol is that remnant iPSCs are a safety concern in moving iPSC therapies into patients. Therefore, we want to remove the iPSCs, which are TRA1-60 positive, at the end of the differentiation process. So here, that was performed using the CGX10 Cell Isolation System. So what you can see from histograms in the upper left, pre-sort, there's a substantial population in the upper left of TRA160 cells, and post-sort, we see that they are greatly reduced. When looking at the cardiac markers, VCAM and CTNT, we see that prior to sorting, they are under 20%, and both of them are expanded nearly 5-fold to 80% VCAM and CTNT positive. We also see a reduction from 10% TRA1-60 cells down to sub 1% abundance post-sorting. So what we can conclude from this experiment is that iPSCs differentiated into cardiac cells stained highly for VCAM and CTNT, and we find that the sorting process enriched these cells and significantly reduced the parent iPSCs, the TRA160 positive population. Last, I want to talk about some new features on the Sony CGX10. A new feature that is available with the system now is remote viewing through virtual network connection. The CGX10 screen can be viewed remotely by users in real time by a wired computer. Due to 21 CFR Part 11 compliance requirements, we cannot remotely operate the system. However, the flow cytometry expert can be positioned outside of the clean room, minimizing the number of personnel that need to be in that space, but still offer advice and direction for the workers in the GMP clean room. Some recommended applications, in addition to what we've talked about today, are for cell therapies, CAR-T and CAR-NK, CAR-Treg in tumor-infiltrating lymphocytes for solid tumors, gene therapy, sorting cells which have been modified with CRISPR-Cas9, and various stem cell therapy workflows. Next, we'll watch a short video summarizing the sorting process of the CGX10 Cell Isolation System.
The CGX10 Cell Isolation System from Sony is designed to deliver multiparametric cell sorting in a fully closed format. A key component of the instrument is an innovative microfluidic chip that is integrated into the system consumable kit. In this video, we will introduce you to the process of fully closed cell sorting taking place inside the microfluidic chip. Here is the overview of the fully closed fluidics design of the CGX10 Cell Isolation System. Once the single-use kit is installed on the CGX10 system, the first step is fluidics priming. In this step, the collection buffer is allowed to flow through the system. Next, the sheath buffer automatically flows through the entire system to prevent bubble formation or entrapment and is drained into the waste bag. The alignment of the chip is achieved automatically by the system by looking at an image of the fluidics channel within the microfluidic chip using a vision system. Following sample introduction, the final optimizations will be completed. As the sample is introduced, the microfluidic chip position is adjusted automatically by the system to ensure high optical signal quality. At this point, the system is ready for data collection. As the sample is flowing, the optic system detects the cells of interest, and the sort delay timing and other parameters are adjusted to ensure the collection of cells matches the desired outcome of high purity at high yields. Clogs that may be formed during sample flow are automatically detected through the scatter-based detection of the sorted sample. Once a clog is detected, the system automatically starts breaking the clog by controlling the fluidics, making multiple adjustments as needed. After the system is calibrated, the sample is hydrodynamically sorted into the collection bag based on sorting criteria. Negative pressure is created based on sort calibration parameters, and the sample is aspirated into the sort chamber. Finally, the sorted cells are collected into the collection bag, placed inside the temperature-controlled cabinet. This is how, with the unique microfluidic chip, the CGX10 instrument enables fully closed, high-speed, high-purity, multiparametric fluorescence-based cell sorting for all kinds of cell therapy manufacturing workflows.
And picking up again, the CGX10 Cell Isolation System is a unique closed cell sorting instrument with customized single-unit disposables and a GMP-compatible user interface. It is ideal for high-speed isolation of highly pure and viable T, Treg, iPSC, and NK cells exhibiting different immunological markers. It can be integrated with closed upstream and downstream CAR-T process steps and has the potential to be integrated into a variety of immunotherapy, stem cell, and gene therapy manufacturing pipelines. Last, I want to invite you to come on our website and take a look at our new application note covering how a Vectis Soliport technology for cell transfection can be integrated with Sony's CGX10 Cell Isolation System seamlessly. Soliport transfected cells that were isolated using the CGX10 system to yield highly cytotoxic CAR-T cells. You can also scan the QR code available here and look at the 3 protocols described today in further detail with our Treg therapy application note, CAR-T therapy application note, and iPSC therapy application note. Thank you.
Thank you, Brian, for your informative presentation. Joining us for the live Q&A, we have Aditi Singh, Senior Product Manager at Sony Biotechnology for CGX10 Systems. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit, and we'll answer as many of your questions as we have time for. First question, asks, how many lasers and how many parameters can be used with the CGX10 system?
Thanks, Casey. Hello, everyone. I'm Aditi, and maybe I can take that question. So, with CGX10 system, we offer 4 lasers and 2 scatter channels. So, we have 405 nanometer violet laser, we have a 488 nanometer blue laser, we have a 561 nanometer blue green laser, and a 638 nanometer red laser. We also have forward scatter and back scatter with the CGX10 system. So the total number of parameters which could be run on the CGX10 system are 10. And that includes 8 fluorescence and 2 scatter parameters. I hope I was able to answer your question. Thank you.
Wonderful. Thank you, Aditi. Okay, moving on. Our next question asks, what is the total running time possible with the standard issue kit?
Hello, this is Brian speaking. With the standard consumables ordered for the CGX10, which includes a 5-liter sheath bag and 1-liter collection fluid bag, the system can be run for up to 8 hours. With a larger sheath bag, it runs up to 10.
All right, excellent. Thank you, Brian. Moving along here, our next question says, please comment on viability of cells after CGX10 sorting.
Maybe I can take that. So as discussed in the talk, we have tried CGX10 system for wide range of applications like CAR-T, CAR-Treg, iPSC-derived stem cell therapies. We have also utilized CGX10 system for isolating NK cells, hematopoietic stem cells, and mesenchymal stem cells. And for all these applications, we have obtained a viability of 99% or higher as the sorting on the CGX10 system, which is based on hydrodynamic technology as shown in our video, is very gentle on cells. Thank you.
Excellent, thank you. Okay, our next question asks, what can be done if there are issues during an OP run? Can you rescue the sample in the user guide?
Hi, great question. So if there is an error during an OP mode sort, the system will prompt the user through a number of steps which are able to continue to keep the sample sterile, disconnecting the sample vessel and the collection vessel, and allowing you to weld it on to another manifold. The system also has a number of internal steps that can be incorporated into your protocol to mitigate any sort of problems. For example, it can be set up to have automatic clog detection to deal with any clogs with samples that are more prone to it. You can also have the system run automatic back flushing on a time-based basis to head off clumping before it can even happen.
Okay, great. Thank you. We have a couple more questions that have come in. Our next one here asks, does the CGX10 provide IQ or OQ and vendor qualification support?
Thanks, Casey. Maybe I can take that. So yes, we do offer comprehensive service package with the CGX10 system for enabling seamless operation in a GMP setting. And that includes preventive maintenance, IQ, OQ, installation qualification or operation qualification, and also vendor qualification. So we do offer all kinds of support, all kinds of regulatory and quality support documentation so that one can take their therapies from process development to clinical or commercial manufacturing smoothly. Thank you.
Thank you. Okay, our next question here asks, does CGX10 offer the ability to retrieve and sort again the negative fraction unsorted cells? And do you have that feature in CGX10?
All right, I'll take a crack at this one. So the unsorted fraction in the CGX10 is collected in a bag, unrefrigerated at the bottom of the instrument. For molecular and analytical assays, this fraction can be recovered using either centrifugation or a syringe to take a small fraction, for example, for RNA sequencing. It is not typically used in a resorting. One would have to spin the entire volume down. The CGX10 is designed to not require multiple successive sortings to obtain all the cells from a negative fraction. The system can be set up on flow rate, concentration, and running parameter to collect a higher than average enrichment if that is needed.
Excellent. Thank you, Brian. All right, our next question here asks, what makes CGX10 consumable GMP ready? Are they irradiated? Sorry.
Maybe I can take that. Thanks, Casey. And that's a very insightful question. So, actually, the CGX10 consumables like the tubing kits used for process development or GMP operation are not irradiated. But in fact, they are sterilized using ethylene oxide gas according to ISO guidelines, which are relevant for typical GMP grade setting. And this ethylene oxide gas method is much gentler than gamma irradiation and provides equivalent sterility assurance level SAL of 10 raised to power -6, which is what gamma irradiation achieves as well. So yes, CGX10 offers equivalent sterility as gamma irradiation, but the method which we use is much more gentler. It's not gamma irradiation, it's just ethylene oxide gas sterilization method in compliance with the relevant ISO guidelines. Thank you.
Thank you, Aditi. Our next question here asks, what are the buffers provided with CGX10?
All right, I'll take this one. So for standard issue with CGX10, it includes a 5-liter bag of sheath fluid, as well as a 1-liter bag, which is more than sufficient for even a 10-hour run of collection buffer. The collection buffer can be supplemented with nutrients, with peptides, or with pluronic to enhance the flow. It can also be substituted with media with appropriate serum concentrations if you wish to collect your cells directly into media.
All right, wonderful. Our next question here asks, how do you perform instrument QC and ensure that instrument is functioning properly?
All right. As part of the CGX10 workflow overall, in addition to the IQ/OQ, which are performed by Sony engineers, there is a QC that can be performed on the instrument using the SUD kit. We supply the kits as well as the 2 bead types required. The total time is about 45 minutes. It can be done in both process developer as well as operator modes, and the beads are appropriately sterilized for a GMP environment.
Wonderful. And we have time for one more question here. This last question that we have asks, how do you perform instrument QC and ensure that instrument is functioning properly?
Oh, I think that was just answered.
Yep, you're right. I'm so sorry. That looks like it was a duplicate. So I'll go ahead and wrap up there. I want to thank you, Brian, for your time today and your important research, and thank you, Aditi, for joining us for the live Q&A session. We would also like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you, everyone.
Thank you, everyone, for attending.